Vegetative propagation is the most widespread means of plant production used in commercial horticulture. Its main advantages are the raising of plants that are identical to the parent plant, the speeding up of plant growth and development, increased plant production, and greater cost efficiency. When we propagate plants by vegetative means, we harvest cuttings and other plant parts, excluding seeds, directly from the plants themselves. This ensures that the new plants created by this method possess all the ornamental or other characteristics for which the plant is notable. This is a crucial facility for nurserymen and indeed any propagator, as it guarantees the correct nomenclature and form of the plant material in which they trade. So, cuttings can be taken from what are termed mother plants. These plants can be especially cultivated out of doors or held in containers in glass houses, polythene houses, or other protected cropping structures. The cuttings are best removed from young, vigorously growing stock. Always make sure that the plant material for rooting is true to name and displays the form of the described plant. Although some propagation material can be removed from young nursery plants, nevertheless it is advisable to set aside specially planted and cultivated mother stock for the annual cutting harvest. The plants should be pruned with care so they retain their vegetative and juvenile condition. The normal weed control and feeding program should be carried out on these specialized stock beds. When inserting cuttings for propagation, there's a choice of materials available. Bear in mind that these must have certain physical and chemical properties, like ability to retain water and air, adequate drainage, as well as being sterile. The type of peat that we most commonly use is medium-grade moss peat. As it is only slightly decomposed, it retains much of its original sphagnum structure. It is a uniform product, sterile, aerated, and capable of retaining water. Nevertheless, for many plant types, the addition of other media makes for better rooting of cuttings. For example, sand, perlite, and vermiculite. Peat is also free of pests, diseases, and usually of weeds. And cuttings rooted in mixes containing peat transplant well, since the material adheres to the roots and forms a good root ball. Peat-based composts have a high capacity to retain water, and when preparations are being made prior to cutting insertion, check by squeezing a handful of the compost. If water runs from the mass freely, it is too wet. Aim to use a compost only when it is moist rather than wet. Once the compost has been mixed, we can fill it into multi-celled trays. As an alternative, we can use rooting plugs. They come in different sizes, and once again an individual cutting is placed in each for propagation. Now, one of the aids to rooting is the use of root-promoting substances. These come in powder form or in liquid form and are applied to the base of the cutting prior to its insertion in the rooting compost. They're then placed in the cell units, pre-dibbled, because the removal of the hormone would occur if you push them directly into the compost without dibbling. Some species that are more difficult to root are better treated with a liquid hormone of a concentrated level. The bases of the cuttings are inserted in the liquid for a second or two before being placed in the propagation tray. The tools that we use for propagation should be of good quality and capable of taking a good edge. They will be expensive, but the investment is well worthwhile because you'll have them for a long time in your propagation career. Keep the propagating knives and secateurs sharp. Secateurs and scissors should also be kept sharp. Propagators must take precautions against wilting of cuttings, especially with spring and summer material. Mist propagation neatly provides the necessary conditions. With this system, sprays of fine water droplets discharge through pressure apparatus and coat the cuttings with moisture. Cuttings will remain turgid and composts moist. The frequency and duration of spray surges are controlled by electronic devices which mimic leaf moisture levels and operate the on-off sequences. Mist is most commonly used from March through September. Although not essential for all year-round rooting, it is important that the base heat facility is available for use during propagation sessions in spring and summer, especially during unexpected cooler periods. A convenient method for providing heat is to install soil warming cables with thermostatic controls. These are laid on a grit or sand base in lines 7.5 centimeters apart. Place wire mesh over the cables to protect during maintenance and to spread the heat evenly. The base temperatures in propagation beds should be maintained at 10 degrees centigrade during winter propagation, that is October through February, and from mid-February onwards it should be raised to 15 to 20 degrees. This is a propagation system which, like mist, maintains high humidity in the space around the cuttings and prevents wilting. Trays of cuttings are laid out on the propagation beds, which are insulated at the base and sides with polystyrene sheets 5 centimeters thick. Polythene film of 20 to 25 microns is placed directly on the cuttings and its edges sealed with light weights. A fully saturated atmosphere is thus created in the cutting space and moisture loss from them is prevented. In these conditions, with a well-managed temperature regime, the prospects for successful rooting are enhanced. As an alternative, the plastic can be kept clear of the cuttings by means of hoops. In systems of propagation where polythene covers are in use, it must be emphasized that shading and ventilation be used to counteract rising temperatures in the beds, especially from March through September. If direct sunlight shines on the plastic, high temperatures soon build up. Even the low February sun can induce temperatures of 25 degrees centigrade or more under the plastic, so as soon as the bright days come, it is necessary to apply light shading to the glasshouse. A thermometer set among the cuttings is strongly recommended to warn when conditions are becoming too warm, and in addition, apply a supplementary proprietary greenhouse whitewash to the outside of the structure. Once cuttings are rooted, they should be hardened off. This operation is best carried out during still, overcast weather conditions. It is recommended that the rooted material is only gradually exposed to the prevailing conditions in the propagation structure over a number of days and nights. Do not carry out the hardening off operation too rapidly, otherwise serious leaf drying out can follow. Once the weaning is complete, Move the rooting material on to the next cultivation phase. Softwood cuttings consist of new season shoots, whether naturally produced or forced under the range of protective greenhouse environments. They are removed from the mother plant by cutting through the base of the cutting at the junction where the new shoot and parent stem join up. The cuttings are typically non-woody, and a cut through this junction with a sharp knife or blade will be through slightly firmer tissue called the pulvinus. These juvenile cuttings are removed from mostly deciduous woody plants and from herbaceous perennials, and being of soft tissue are very susceptible to wilt. So every precaution to maintain their turgidity should be taken by the provision of shading during sunny spells, and great care must be taken during the operation of weaning. Greenwood or semi-ripe cuttings are at a later stage of development than softwoods, and the shoots of the plants are beginning to firm up. This is usually in late spring and early summer, and often are taken as nodal as well as tip cuttings. Rooting of the more difficult species can be encouraged by lightly wounding the bases. As with softwoods, the atmosphere surrounding the cutting should be one of high humidity, whether it be mist or warm bench and plastic methods. Care should be taken to apply appropriate shading to this summer propagation material. Semi-hardwood cuttings are propagated at a later stage again, when the tissue is partially woody, and these are taken from midsummer onwards. They will have a length of 8 to 15 centimeters, are usually wounded at their bases and taken as nodal cuttings. These shoots of the mother plants will have lengthened during the summer, and often 2 or 3 cuttings can be got from the same stem. However, avoid the use of very woody stem bases. Branches of conifers can be gathered during the October to February period. Semi-hardwood cuttings are removed from these, about 10 centimeters in length. Basal leaves are removed, rooting hormone applied, and cuttings are inserted in multicell trays or individual plugs. The cuttings are propagated in the warm bench and plastic system, with base temperatures being held at 10 degrees centigrade. These propagation conditions are equally suitable for an extensive range of broadleafed evergreens. The usual care should be taken of October cuttings for fear of unexpectedly hot October days, and also from mid-February onwards. Leafboard cuttings or mallet cuttings. These types of cuttings are often used in propagation material which is in short supply, as in the case of camellia, or where the growth habit of the mother bush dictates such cutting types, as in Mahonia. Always select vigorous shoots which have well-developed buds. The cutting consists of a piece of main stem cut above a bud and above the next bud down. Continue this operation down the stem, and the resultant pieces will consist of a stem piece and a bud in the axil of the single leaf, and the stem should be wounded. No rooting hormone is necessary, as it may affect ultimate bud growth. Insert the cuttings in the tray cells, with leaves facing in the same direction to facilitate spacing. Hardwood cuttings. These cuttings are woody and are best taken after leaves have fallen in the case of deciduous mother plants. They have high levels of food reserves, and by this means can survive the short, dark days of the dormant season before showing rooting in the spring. The tips of these stems should be removed. The top of the cutting is cut above a node and the bottom below a node. The finished article should be of pencil thickness, 20 to 22 centimeters long. Wounding is an advantage, as is the application of rooting hormone powder. Bundle the cuttings in 25s using polypropylene twine and insert to 2/3 of their depth in a well-drained medium. Propagate in a cool greenhouse. Base heat would help early rooting. Keep the cuttings moist during the winter, but avoid conditions which encourage bud break of the sticks, as this will severely hamper rooting. If hardwood cuttings are propagated out of doors, make sure the site is protected from winds and has a good, well-drained soil. Recent practices of inserting individual cuttings in propagation plugs have shown encouraging results. Root cuttings. The plants which are usually suited to this method of propagation are those which naturally throw suckers from their bases, and these include Aralia, hibiscus, Russ, or Campsis. The material is prepared between November and March, and 1-year-old roots are ideal. Very thin roots lack food reserves. The more substantial roots are cut up into 6 to 8 centimeter pieces. Cross cuts are made in the pieces next to the plant crown, and slanted cuts at the other end. Treat the root pieces with a fungicidal powder and insert the slanted end of the cuttings upright in pots. Cover lightly with sand. Sprinkle the thinner pieces on filled trays and cover with a layer of compost. Place on a greenhouse floor or in a cold frame and keep frost-free. Layering. This is an operation in which shoots still attached to the mother plants are layered in the ground in order to produce roots. Once rooted, usually in the following autumn, the shoots are cut away from the mother plant and cultivated as individuals. Layering is suitable for difficult-to-root subjects such as acer, magnolia, and cotinus. This method of propagation has a high success rate, though much room and attention is needed for the creation of commercial-style layer beds. Some species take up to 2 years to produce satisfactory plants from layering. Now, simple layering is mostly done during the May to July period when active growth has ceased. Shoots are wounded, bent, and pegged into soil. The above-ground part of the shoot is staked for stability. The stem part in the ground is covered with soil and kept moist. At the end of the season, or when the shoot is firmly rooted, cut the new plant from the mother plant and cultivate it separately. Serpentine layering. Species which produce long annual shoots, for example Wisteria or Aristolochia, are suited to this method of propagation. The stems are brought to ground level, buried at intervals, so that each above-ground part has a healthy bud. Roots will form on the buried sections, which can be cut into separate pieces to form new plants. French layering. Viburnum and cotinus are frequently produced by this method. The dormant stems are pegged down in the early part of the year. When the young shoots along the stems reach 10 centimeters in height, they can be gently covered with soil. As soon as they are growing strongly again, they can be earthed up once more. When leaves have fallen in the autumn, section the plants and cultivate individually. Stooling. This is a method of layering mainly used by fruit rootstock producers. Plant mother stalk in the autumn, and when it's well established in the following winter, cut the stalks back to a stump. The shoots which develop from these stumps are earthed up during the growing season. After the young shoots have shed their leaves, cut and lift the new plants. Cutting this plant yield from the mother stalk prepares the plant for further shoot production in the following season. Division is the simplest method of plant propagation, whereby the plant is broken into small, separate units. The plant organs suitable for division include rhizomes, for example, iris, lily of the valley, or runners above and below the ground, for example, ajuga and strawberry, or else multi-stalked or multi-stemmed shoots, erica or vinca. Springtime is the best period for division of the herbaceous and woody ornamentals, but do so before the end of May. Autumn division is suitable for astilbe and peony. These die back naturally then, and are lifted and dried off, kept in a frost-free area, and planted out in spring. Bulb propagation from scales, for example lilies. Dig the bulbs from the ground 8 weeks after blooming. Clean off surface dirt without getting the bulbs wet. Peel the scales off the bulbs, breaking them at the base or cutting them away with a sharp, sterilized knife. When removing the scales, take a small piece of the basal plate at the bottom of the bulb. Treat the bulbs with an antifungal powder and also with rooting hormone powder. Mix the scales with damp vermiculite in a plastic bag and place in a warm, dark place for 3 months. At the end of this period, place the scales in a refrigerator at 3 or 4 degrees for about 2 to 3 months, and plant out after they begin to sprout.
Micropropagation is used by specialist laboratories to produce plants in a sterile environment in which multiple shoots are produced from single buds. And this micropropagation system can be applied to a very wide range of plants, from herbaceous plants, from trees and shrubs, and even bulbs. The system that we employ in micropropagation involves culturing buds on a medium that stimulates the growth of the buds to produce a mass of shoots. And we do that by providing nutrients that the plant bud needs, all the major nutrients, nitrogen, phosphorus, potassium, micronutrients, all the trace elements, vitamins, and importantly, we also have to add in growth regulators and sugars. The growth regulators stimulate the development of shoots from the buds, and once the shoots are produced, we then dissect them apart into individual shoots, and then we transfer them to a different culture medium containing hormones that stimulate rooting. And once the roots are formed on the shoots, they're then more or less an independent plant, and they can be transferred to the greenhouse. But in the greenhouse, we need to acclimatize the plant to the lower environment in the greenhouse. So that's done by gradually reducing the humidity in the greenhouse. Micropropagation is used, as I mentioned, for a very wide range of plants. It's usually done in commercial laboratories on a fairly big scale, in which usually tens of thousands of individual varieties are produced.
Production on a commercial scale. Many of the hand movements in the operation of propagating plants from cuttings can be carried out automatically. Here is the machine removal of softwood cutting material of spirea. The coarse cutting material is gathered, transferred to the preparation house, where clean cut material is prepared for sticking. The availability of cold store facilities is important during dealing with large numbers of cuttings. Every care should be taken in preventing young softwoods from overheating during the stages of harvesting and preparation. Amongst the large numbers of manual operations inherent in a successful plant propagation program, there will always be opportunities for efficiencies. Some easy-to-root subjects can be suited to automation in cutting harvesting, but nevertheless will still require the skilled attention of dedicated propagators. As always with softwood and semi-ripe material, the availability of cold room facilities will provide the safeguards in preserving good cutting condition before being transferred to a suitable rooting environment. The aim at all times is to maximize rooting numbers, and the provision of favorable rooting conditions will help in reaching this goal. The combination of personal skills and scientific principles are applied in the operation of plant propagation. As the propagator gains experience in dealing with these principles, the solid foundations of a successful annual propagation program will be firmly established.
Pastel moon, red watercolor sky, the perfect way to end my day.